Yes, I hope you can see my screen as well.
Yes, sir.
Yes, okay, good. So in the last class, we discussed something very general and actually very important to each of us. That is about ethics, not just about a profession, but also as a part of general living as well. I hope you have thought about it or engaged in some discussions with your friends, family members, because it is all about trying to understand different diverse people, and then getting diverse opinions, and then trying to see what is most rational in that given conditions, and how we can probably develop the skills in thinking about what is right and what to do, and so on. So before a class prior to that, we discussed about primer designing and other things. So I would like to still use that right now and try to using primers. Today's class is we are trying to develop or trying to take an analytical approach, okay, for based on PCR. So I want you to remember this, that when you are going into science, I would like that the first is we should be able to have skills in some memory, remembering, and then we would need understanding of the concept, the basic biology, and so on. And then we are now, as the recombinant DNA technology is an applied science, we should be able to take it, apply it, and um, analyze or design new things or create, right? So we want in this course, if you look at the outcomes as well, we want that each of you have an ability to apply what you have learned so far in the basic courses, and that's why I would want to spend a little bit time. Today's class is about that, and I will try to use PCR based methods and so on. So first thing is, what is meant by homozygous and heterozygous, right? So if there is a gene A, say for example gene A, and gene A could be having alleles called as A1, A2, A3, and so on. What are alleles? Alleles are copies of the same gene, except that there is some sequence difference. When you say sequence difference, what could it be? Sequence difference, it could be that one gene is, say for example, if A1 is this way, A2 could be longer, right? A2 could be longer because there is a kind of insertion or so. Assume that there is an insertion here, and that's why it is longer. You could also have otherwise it's something like A3 could be having a deletion, okay? But I will not take that right now. But what it means is, I'm trying to say A and A3 are almost same, except there is a small difference, and that difference is, one second, there is a little bit. So A3 has same size. Say for example, this is 300 base pairs, and this is 400 base pairs because there is an insertion of 100, and then this is 300 base pairs. Between A1 and A3, what is the difference? There could be a single nucleotide polymorphism. which means there is a point mutation. That means in that particular base, there is a difference in the sequence. Say, for example, if there are two sequences, A, B, okay, I'll write the nucleotide bases. That's better. Say, for example, if there is a sequence G, T, C, A, T, G, and its complementary sequence would be something like this. And assume the allele, this is assume allele A1, and A3 could be having something like CTAG. The difference between A1 and A2 is in this position number 4. Okay, I'm just assume that I picked up a small part of it. In this position, there is nucleotide polymorphism, in the sense there is a change in that sequence, right? Those are called as SNP. You must have read it long time ago in mutations, molecular genetics, and so on. Single nucleotide polymorphism. In today's class, I hope you remember, you know that in the syllabus we have ways of molecular diagnostics, right? That means you want to find out if there are any insertions, deletions, or if there are any mutations. I mean, we already have the prior knowledge of about the sequence of the or the nature of the change, whether it is insertion, deletion, or a single nucleotide polymorphism. And we will have to determine, a patient sample comes, and we should be able to determine if that person has that mutation or not. In that case, and if we should also try to know if that person is homozygous or heterozygous for a particular mutation or not. In that case, how do we approach? And the goal today, as I said, is based on primers or PCR-based methods. Because we discussed about primers in the previous class, it would make good enough sense. And I also want you to develop an mindset for analysis and interpretation. Because when we study the next part, that is about vectors, we will be constructing some things. We'll be constructing genomic libraries, maybe, or some kind of selection and so on. For this, we should develop these interpretational skills. 1 second, I just want to ensure we are recording. Yes. So first thing we will see is how to differentiate, how to use it for analysis A and A1. A1 and A2. A1 is about 300 base pairs, and A2 is about 400 base pairs, because there is an insertion of 100 base pairs. Okay, so I'll try to use in the same space so that hopefully we'll be able to do it fine. Let's see. Say, for example, there is the DNA is something like this. We say gene, and here too, this is double-stranded DNA. And I'm putting it also in different ways. I'm not going to give you complete details. I'm only going to give you as many details required for you, so that you will be able to solve it as if it is a puzzle. It's not very complicated, of course. But if there are problems, we will then try to describe it properly. So this is 100 base pairs insertion in A2. So we have A1 and A2. Say, for example, a person, we got a sample. From the sample, we isolate DNA. With the DNA, we will do PCR. Okay. The PCR will have, for PCR, we need to have primers. And the primers are designed in such a way that the one primer will be attached this way. Okay, that green one showing, and the arrowhead, half arrow, is showing the direction of the primers, 5' to 3'. So polymerization would happen in this way, and in here it would happen in this way. So the same primers, remember that A1 and A2, the sequence are absolutely same. So the same primers, we will call it as forward F and reverse R. The same primers can also recognize the A2 allele as well, because the sequence, the rest of the sequence is same between A1 and A2. Only the middle part is different. So if we have performed PCR, we are likely to get, what are we likely to get? That's the question I'll have. So let's see, let's try to solve it as if it is a puzzle and see what we will get. Say now I'm going to give you the question. I have taken, this is the ladder, and you have done PCR, and then you're going to do electrophoresis. You have done PCR with F and R. And here is the, there are 3 bands. So this is, say, 100, 200, 300, 400, and 500, the ladder. Okay, I mean, I hope you remember from your molecular genetics one. So if I perform PCR, and if the sample has given a result like this, there is only one band, what does that mean? What does that mean about the sample, about the genotype? Do you think the person has 2 copies of A1 or 1 copy of A1 and A2? What could be the possibilities? Anybody? What does it mean if they get 1 band of 300 and 1 band of 400? And what does it mean if they get only 1 400 band? Anybody? Can you hear me?
Yes, sir.
Okay. Are you trying to answer?
Yes, I'm thinking of the answer, sir. Yes.
Okay. Good. Okay. If an individual had A1 and is homozygous for A1, that person will have a DNA of this length. I assume that each is 2 strands. And if PCR is performed, one of the allele will give about 300, and the other one will give 300. Right? So both will give only 300. They will travel together, and you would only get 300 band. You will not be able to see a 400 kV or 400 base pair band. That means both alleles must have been A1 and A1. The sample from A, for example, this
Sir,
sample. Yes, please.
sir, that's the case. B sample might have an allele of A1 and A2 because A1 is 300. A2, let's say, for example, it might be 400.
That's right. Absolutely. Okay. And A3 is, sorry, the sample C would be?
Just A2 and A2.
Yes, homozygous for allele
Homozygous.
A2. Yes. Okay, it is simple, straightforward, no issues at all. So in CA exams or anywhere, usually I ask about these questions. The questions will be like these: either data, a gel pattern might be given explaining all the experiment first, and a diagram could be given, and you are asked to interpret the results. Or you would be given a data or interpretation, would might be asked to draw a gel diagram, and then each of you should be able to do that. As I said, this course is about applying what we have already learned. So we should do those things and try to develop those skills. So if you have any questions, if you didn't understand, you please always contact your friends or me for help. This will be very useful when you discuss with others, actually. So I hope you understood how we could try to find out if the interpretation can be done based on size, and using PCR we are able to determine if a sample is homozygous or heterozygous for a particular mutation also. Okay, of course we should have had prior knowledge about the mutation or the insertion. We should have had prior knowledge of the sequence so that we could design the primers appropriately. Okay, so the next one is about how assume that if it is A1 and A3 alleles, the size is same, only the difference is only one nucleotide. Somewhere one nucleotide is different. Before going into that, I will try to quickly review the primer designing. In this case, the primers are only, I'm just trying to quickly review. So for primers, something very similar to that of cloning primers. So I'm just putting this way, and we have, say, sequence GACT, and this is the rest of the DNA. T. Okay, one second. And C. This is C. CTAG. And CTGA. This is the DNA, and I want that we design primers. And always, this is 5 prime, and here is 3 prime, and this is 5 prime, and this is 3 prime. Now, if we want to design primers, polymerization always happens from 5 prime to 3 prime direction. Therefore, and in PCR, we would want that the polymerization is converging on both sides like this. That means the primer should be designed in such a way. So I will design primers such a way that they will converge. So it should be TCAG, and this would have been the 5 prime. And the other one would be GATC. This will be the 5 prime, and polymerization would occur this way for the top strand, and this way for the bottom strand. And that's how the products will become the templates for the next round of reaction, and that is the chain reaction. Exponential increase in the number of the products formed. Right? So now I will try to explain the question that we are trying to address: how to differentiate two alleles which differ only in one base, that is single nucleotide polymorphism. So say there is a sequence GATGC, and I'm going to draw another one. So this is the DNA, and TACATG. AC, CTA, CG. And this is the 5 prime, and here is the 3 prime. So basically, there is one problem here, is that there are 2 copies, except there is a problem with one of them. That is, assume that this base, that is the base where there is a difference in the sequence. That is, assume it has TA, right? The rest of the whole sequence in these 2 alleles is same, except in that position, okay? Everything else is same. We, as per the first slide we discussed, this is A1, allele A1, and this is allele A3. Okay. So we are supposed to design primers for both of them. And we will do it in such way that, like in the previous slide that I have described, we will use a primer for this G, A, T, G, and C. This will be the 5 prime. Sorry. 5 prime, and the other primer will be here. I will take C, A, T, G, T, A. Don't worry about the number of bases. On one side I wrote 5, and one side there are 3. Assume that it is how it should be. 5 prime, and the other one is 3 prime. Now we will look at this sequence. I can use the same primer here too, because this sequence is same. Can I use the same sequence here? No. Why is that? There is a mismatch in this one. This is a mismatch. So I should have designed a new primer with the correct sequence. When DNA polymerase is polymerizing, it should want that the initial bases on the 3 prime end should be of the primer should be perfectly matching. So because that is not there here, we have to design a new primer, that is this one, right? So now we have 3 different types of primers. Primer 1, we can call it as P1, P2, and P3. Okay, I'll use green color. So this is P1, primer 1. And this one is P2. And we have a different one, P3. This one, the primer here that is shown here is the same as P1, right? So we don't have to call anything, but it is just P1. Now, think about if a person had A1 allele, A1 and A1. That means that person would have DNA, something like this, and you would have used, if I used P1 and P2 primers, okay, if I use P1 and P2 primers, P1 will bind here, P2 will bind here, and will give a product eventually after PCR, several rounds of PCR, right? Assume based on what we did before, we'll say it is 300 bases. Okay. The person had 2 copies of A1 and A1. So there will be a product. It doesn't matter. If I used a different set of primers, like P3 and P2, what would happen? Well, this primer will bind here. That is fine. Whereas this primer, the P3, will not bind or will not participate in the chain reaction, right? Polymerization would not happen because we are saying the alleles are A1 and A1. So if we use P2, P3 and P1, there will not be a product. If you are lost when I'm trying to describe this, it's all right. I'm going to repeat in case you need it. So don't worry about it. Just we'll go through ahead and we'll try to understand it. And if we have a problem, we'll get back again. So when it is confusing, sometimes it's okay to go ahead so that it becomes clear. Because if it doesn't, we will do it anyway. We will repeat it. So I'm giving like the gel diagram that we have done before, right? So the sample is, we have to have two different, 1 second, I need to draw one more. So I'm going to give 3 samples. Okay. In the 3 samples, I will write to, this is, say, this person is A1, homozygous for A1 allele. The person here, the second sample is heterozygous, A3 and A1. Person 3 is homozygous for A3 allele. And here I have put another box here, right? There I'm trying to give you the primer, what primers have been used. A1 plus 2, okay? P1 and P2 primers. In the other reaction, P2 and P3. The same is done for all of them. 1 plus 2, primer 1 and 2, primer 3 and 2, primer 1 and 2, and primer 3 and 2. Now, assume that the ladder would be something like this, 100, 300, and 600. What can we expect of the bands in A1, A1? If we use P1 and P2, like is shown here, P1 and P2. This is P1, and here is P2. What would we get? Anybody? What bands will we get in this lane? Will P1 and P2 bind to A1? This is the A1, right? Here, I hope you can see it. This is A1. P1 is this, and P2 is this. They will bind, and the PCR product will form. And we said it will be about 300 bases. So
there's a band in 300.
Yes, there will be a band in 300. If I use P2, if I use P3 and P2, what will happen? The genome is homozygous for A1. That means it is this one. And if I add P3 here, there will be a mismatch between this. This is the primer T. It won't bind here, base pair with this penultimate the complementary strand. So DNA polymerase will not be able to make a product. So there will not be any product here. And just to indicate empty space, I'm putting like that. If the sample was A1 and A3, then what would happen? Like in the second case, A1 and A3, what would happen? So in that one, there is A1, and we are using 1 and 2 primers. The template A1 will act as template for P1 and P2, and we will get a product. In the second PCR reaction, we are using P2 and P3, which is P3 is specific for A3 allele, and the person has A3. So therefore, there will be...
There will also be another band.
That's right. There will be another band here like this. And if we see the last one, there's P1 and P2 will not give any product because P1 is specific for A1. right?
There will be a mismatch for A3 as a result, since 1 and 2 won't have that primer sequence as it relates
That's
for
right.
A3.
That's exactly correct. So this C here, a primer, the last of the primer cannot base pair, so they will not be a PCR product. And here we'll have a product. So in this way, we can always make or design the protocols and solve or try to figure out what is a person, what is the heterozygosity, does the person have the mutation that we are looking for or not. Okay, so in the CIA exam, for example, a question could come up and you are supposed to find the answers, interpret the results, and write a small results and discussion, right, explaining why you think that is the answer. Okay, any questions here that were any possibilities of confusion? If you let me know, I'll try to do it again. Anybody has any questions?
Sir, I have a doubt regarding
Yeah.
that bands alone. Like, for
Yes.
example, if sir, what if there is a chance of having two bands of just a 300 and a 600 also? Like, that is it related to the different fragment lengths?
In this case, are you asking in this case?
No, sir. What if there's a change in scenario? For example, like A1 is 300, but instead of A3, instead of 300, it's still say it's 600 or maybe even less than that. So, is it related to the fragment length based on that?
So I don't know. Let me try if I have given three scenarios here: alleles of A, gene A1, A2, and A3. Okay. In that, we are assuming this is normal. This one has 100 base pair insertion, and the A3 has a single nucleotide polymorphism or point mutation. Okay, substitution. Unless there is any other change that we don't know of, and if we are sure we designed the primers in this way, you cannot or should not have other bands. Okay, suppose if we are taking a sample and from a person and we perform PCR, say in this case, and there
Sir, I
may
was just
be,
sir
yes,
also provided assuming that the primers are they do not have a mismatch. Like I
yes,
just
yes,
wanted to mention that also. Like assuming that the primers won't have any mismatch.
yes,
I'm just saying based on the fragment length, it could differ. Like the bands could differ based on the fragment length. That's what my question was. Like I was assuming that the primers were pretty much the same and don't have any mismatch. Like if there's
yes,
a mismatch, then it's obvious that there's no bands. There won't be any bands formed.
that's right. If you have any other bands formed, big or small, that means it is a new allele you found probably. Okay, if you have any other bands, there are also possibilities of misformations of say if the non-specific binding of primers, sometimes that is also a problem when we design primers. If there is relatively a huge match somewhere else, it's improbable, but it is possible. If there is a mismatch and non-specific bands are coming, they should be in all the lanes, right? One can expect that they are all in all the lanes, non-specific bands, because of primer binding somewhere. But if you find somebody having a band like this, when you're performing an analysis with the current knowledge of whatever you have, and you have this band is normal, 300, but this is something new, right? It's out of the expectation. That could be a new allele you might have come across. Okay, I don't know if I addressed your question, but please tell me
Yes,
if.
sir, something like that, sir. Like
Okay.
I was just asking her whether, like, since you've shown that in that very same column we might have three different bands of some different lengths. like one might be which is expected. Even for example, even the length might be even bigger. Like it's for
Yes,
example
it could be
500, 500, it
larger
might be
than
bigger. It
600.
could
It
be
could be,
larger, but
yes,
if as long as is the allele we know the size which is expected, if we know that it's going to be like that, for example 500, and if we get that expected outcome, that's fine. But I mean,
yes,
some of them, if it's unexpected ones, then it's like as you mentioned,
it could
like
be a
the
new
reasons
one.
of the primers may have some non-specific
Yeah,
reactions
if it is
due to some
non-specific,
reasons.
we will find it in all the samples. So if it is in all the samples, usually we don't count it as any specific result. Okay. If there is something special like this one, then there is, yeah, something new probably. If you found something like this is probably an additional copy with a deletion or something similar sequence in somewhere else in the genome. Okay, that will complicate the stuff, but certainly that is a good question in the sense that there will always be new things when we do, say, for 1,000 people. If we do an analysis like this, we will find differences. Our genome is too huge to start with, so the probability is always there. In diagnostic tests, generally, they would try to develop the diagnostic test after thorough understanding of everything or maximum data available. Yet, there are possibilities that X may come, and then further analysis is required to see if it is an extra copy or if it is a deletion or a new allele and so on. Okay? Yes, Ajit?
Yes, sir. Yes, sir. I
Okay.
mean, while we solve some of the related problems like this, then maybe we might understand something.
That's right. That's right. Certainly. Have you done these before? Any of these analysis in genetic engineering or something?
Only a handful of them, like little bit of those. While
Okay.
we were studying that, like that is like I think before doing the that whole cloning experiment, the vectors. I think that was where the first unit. I think somewhere we've started doing these. Only a handful of them
Okay.
we've done. Sir. But
Yeah, we'll
yes.
also do not so many, but everybody should have gained expertise on in interpreting. That will be a part of the assessment. So everybody should try to improve. We have 15 minutes, so I want to give you something else. You know about, I hope you know about epigenetics, right? In epigenetics, DNA methylation is one of the important uh, important aspect or kind of modification that is generally there. I will talk about a little bit about epigenetics later. Right now, let's finish the part, and I will repeat this again in the next class or some other class. Remind me. So say, for example, this is a promoter you have, and there might be a lot of G and C's here, and they might be methylated. Okay. So I'm putting those indicators are methylation. If the promoter region or the operator region is usually if it is methylated, usually expression doesn't happen. Whereas unmethylated one, expression happens. This is the fundamental concept of what it means to be epigenetics. Epigenetic modification, meaning that it is not the change in sequence. If it is a change in sequence, we would call it as a mutation. It is not the change in the base sequence, rather it is a modification of them. So the sequence will remain the same, but there are chemical modifications. Histone modifications are also there. We'll just stick to DNA methylation for this purpose. And is there a way to find out if DNA is methylated or not? Say, for example, we have a sequence like this.
Sir, one way would be if a restriction enzyme, when we cleave a DNA sequence, if it doesn't cleave, then there is a chance that the DNA might be methylated.
That's right. Absolutely right. So if we have a sequence like this, okay, BAMH1, and assume that the C's are methylated on one case. Okay. is having some trouble. I hope I'll fix her. I have to buy a new one. Okay. If we add BAMH1 to both, then it is one of it will be cleaved, this one will be broken, right? Whereas this one will not be cleaved. That is because of the methylation. Okay. We can use restriction modification, restriction endonucleases to figure out the epigenetic status of a DNA. How can we do that is by, I hope you also know about the southern blotting. If not, we will discuss it as a later part. Right now, I just thought I will introduce analytical thinking and probably also primer-based or PCR-based one. Right now, I'm just touching upon restriction endonucleases. Don't worry if it is if you find it complex or difficult at this time. So we can certainly use restriction modification systems to find out whether a DNA is methylated or not. So what I will do right now is I'm giving you a scenario. We are biparental. That means one set comes from maternal allele and one set comes from, that is paternal and the maternal allele. There are concepts in epigenetics like genomic imprinting and etc., etc. In that case, what happens is some genes are imprinted, means something like DNA methylation, for example. That means those genes will not be expressed from the paternal allele, but they will be expressed only from the maternal allele. So in stem cell research or in many epigenetic studies, this is also a way of doing things. It is, of course, associated with southern blotting and so on. We will discuss that later. Right now, let me introduce this. Say, for example, there is a DNA. I'm writing it as single DNA. And I'm only taking this much part. It's a complex one, but just for the sake of understanding the principle, I'll put it this way. And there is another site here. So assume that this is BAMH1 site. This is another BAMH1 site, and this is BAMH1 site. Or I will change it a little bit. Sorry. I'll put this as EcoR1 site. This is EcoR1 site, EcoR1 site, EcoR1 site, and BAMH1 and EcoR1 site. So I'm talking about this as one DNA molecule. This is another DNA molecule, right? If in the second one, in one of the alleles is mutated, is methylated like this, and assume the total length of it is 500. This is 300, and assume this as 400. So the total length is 700. I have given you this scenario. So what will happen if I digested this DNA alone with EcoR1? This is just an introduction, so don't worry.
Sir, meaning that both of these sequences are just cleaved with EcoR1, is it, sir?
Yes.
So just with EcoR1, both of them will produce the equal amount of fragments as...
Tell me, what is it?
Sir?
What will be the size of it? If I digested only with EcoR1, what will I get? We should be getting a band that is 700.
It's only 700, sir. Yes, sir. Because
right. Yes. 300 plus 400. The next one I am cutting with, that will be the last, E plus B. EcoR1 and BAMH1. What bands will it be?
2 bands. One is 300, the other one is 400.
300 and 400. I told you about this, right? This BAMH1 site is methylated.
Methane.
So what do you expect?
So then we'll have one band at 700 because that
That's
won't be
right.
clear.
That's right. If it was giving only bands like this, 300 and 400, what does that mean?
Meaning that one, either it's not methylated or there's a mismatch in primers.
No, no, there are no primers, restriction sites only, right?
I think then it's not methylated.
Yes. This means if you get a result as shown here, that means there is no methylation on the BAMH1 site. If it shows like this, the same reaction you're doing, sample is same, DNA. You have done it everything in the same reaction. You have found that that means one of the strand is getting cleaved, whereas the other, not strand, one of the DNA molecule is getting cleaved, whereas the other molecule is not getting cleaved. And why it is not getting cleaved? It is probably because of, it's most likely because of BAMH1. So BAMH1, I'm GGA TCC. I think DPN1 sequence is for, I'm not too sure, but I will see it like this. CG. So we also learned something about methylation dependency when we were studying about restriction endonucleases. In that, we learned about some enzymes are methylation dependent, which means that they would cleave only if the DNA is methylated. In that restriction site, if that site is methylated, then they will cleave, otherwise not. Such a case is DPN1. If we add EcoR1 plus DPN1, then the sample here should have become, what should it become? Anybody? What brand should it give rise to?
400 and 300. There
Yes,
won't be a 700 band
that's right.
because it's since it's methylated, only when it's methylated it will be cleaved, so there won't be a band at 700.
That's right. So typical experiments will have this way to confirm a methylation, DNA methylation status at that particular site. Okay, they would do something like this with EcoR1 and BAMH1. If there is a band left like this, they would also want to prove that by addition of a methylation dependent or insensitive ones, there are enzymes which are insensitive ones, they would cleave. Okay, I made a mistake here. Can anybody find that? So if I had, I will put as insensitive one. Okay, that means assume that the sequence is same as GGATCC as BAMH1, but it is insensitive. That means that what I wrote there will cleave the site irrespective of methylation. In that case, it will give two bands like this, right? For DPN1, what did I tell DPN1 is? DPN1 is methylation dependent. That means it will only cleave if there is methylation. It will not cleave otherwise. So what should you get then? This strand will be cleaved by DPN1 because that site, what I said, the same sequence, assume that same sequence specificity. It will cleave the methylated strand, but will not cleave the unmethylated one here. So that will remain like this. This is one of the very
Yes, sir. I
interesting.
spotted that when I was explaining that for the second one. I didn't
Okay,
include the first name. Otherwise, it would have not been methylated. I mean, if it's not methylated, anyway, it won't have been cleaved. So there would have
that's
been a
right.
band at 700.
That's right. Good. So here you have to remember methylation dependency. This one is methylation insensitive. These are theoretical at this point, okay? But I hope you get the
Sir, so
you get the idea. Say,
regardless of its methylations or not, it will cleave either ways. It
yes,
won't.
insensitive ones.
So, like, are there any enzymes for that? Like,
Yeah,
is it?
yeah. Right now, I can't remember. Say HPA2, and there are sites actually. But
Okay,
what
sir.
I have taken here are theoretical. This is
Okay,
assuming
sir.
I have taken BAMH1, I wrote instance 2, and DPN1 I was saying. This is assuming that they are all going to recognize similar sequence, GGA, TCC. This is theoretically, okay? But the concept is, if this one, the BAMH1 will cleave only if unmethylated. Insensitive one that I said will cleave both, methylated or unmethylated, it will cleave. The dependent one will cleave only the methylated site. Based on that, we will get a gel pattern. And looking at the pattern, we should be able to identify what has, what is the status, epigenetic status or methylation status of that DNA. Okay? Don't worry, we will discuss it again in case you have any issues, or either way, we will do that. I hope I want you to watch this video over again, because from now on, this will become the tone of the rest of the lectures, because we want to discuss analysis, we want to discuss interpretation, um, we want to discuss creating and designing new genetic elements and so on. So please have a look at this um, and come back for the next class. Okay? Any questions? Anybody?
Sir, so
Yes.
since based on these patterns in terms of insensitivity or methylation dependency or some of those, if it's given in question,
Yes.
so and also based on the band, so in that way we can relate the heritancy of if they've given any individual sequences. Based on that, we can compare the relations.
You meant something like paternity issues, or
Maybe
or
related to that, but not exactly that. I'm saying
no?
like
In
in
this particular example that is visible right now is about epigenetic status. So I need to also describe a little bit about epigenetics in a later class, or I'll share you a video which you might want to watch it.
sure,
Okay.
sir. Yes, sir.
And I want to know that has everyone took stem cell biology? Are there anybody who hasn't studied stem cell biology?
Sir, I didn't
Anyway,
take stem
okay,
cells
okay,
already.
that's all right. We will have an introduction to epigenetics somewhere, and something about genomic imprinting. I would share videos with you probably, and we can also have a discussion on that, and then how to analyze it. Okay?
Sure, sir.
Yes. Thank you very much, and we'll meet in the next class.